today's review is everybody's favorite stuff, chemistry, or our anchor is called Chemical Basis for Life. And if you remember back to what we talked about some basic stuff. We first started with the atom and the basic idea behind what everything is made up of. And anything that takes up space and has mass is what we consider matter. So this can be living things, this can be non-living things, and no matter what type of matter we're talking about, everything is made up of molecules. And specifically with living things, we talked about organic molecules or organic compounds. And all of those compounds are made up of atoms. And atoms are those basic building blocks that make up everything. And if you remember what an atom actually looks like, remember we have a central region called the nucleus. And in the nucleus we have little particles called protons that are positively charged. And we also have little particles in the nucleus called neutrons that are neutrally charged. And outside of the nucleus we have energy levels where our electrons are held. And electrons are negatively charged. And typically in an atom, the first energy shell can only hold 2 electrons, and the outer shells can hold up to 8. And if you remember our magic number, in order for an atom to be stable or happy, the first shell is filled when there are 2 electrons in there. Any less than 2, and the atom would like to get another one. In the outer shells, our magic number was 8, and if an atom has less than 8 electrons out here in the outer shell, it's unstable, it's unhappy, and wants to get more electrons. This leads us to our next topic of chemical bonding. In order for atoms to become stable or happy, they make sure that their electron shells are filled. And there are 2 different ways that atoms can do this. They can either share electrons. So for example, between the hydrogen and the oxygen of water, they share their electrons so that all of the atoms are happy and stable. If you look in this finished water molecule, the hydrogen atom has 2 electrons in its first shell now. Previous to the bond, it only had 1 electron. So it went from being unstable to stable because it's sharing an electron with oxygen. Oxygen only had 6 electrons in its outer shell. So in order for it to be happy, it's sharing electrons with 2 hydrogen, thus H2O. And again, the main idea behind this is that covalent bonds are when 2 or more atoms share electrons in order to be stable. And the magic numbers play a role in that. The second type of chemical bond are ionic bonds. And this happens when we have one atom stealing an electron from another atom. And that's the case in table salt or sodium chloride. Sodium only has 1 electron in its outer shell, so it's definitely unstable. And chlorine has 7 electrons in its outer shell. Chlorine is so strong of an atom that it pulls this electron away from sodium. And because chlorine is gaining an extra electron, it's gaining a negative charge. And because sodium has lost an electron, it's lost the negative charge, so it ends up getting a positive charge. Remember what we talked about with opposites. Opposites attract. So because the sodium has a positive charge and the chlorine has a negative charge, these 2 atoms attract to each other and result in an ionic bond. If we look at these 2 different types, the main ideas are that bonds hold atoms together, as in water or as in salt, and they result because of what's going on with the electrons, trying to make the atoms stable. Now that you've refreshed your memory on some basic chemistry like the atom and how atoms hook together, let's focus now on chemistry in biology. And we're going to look at two topics. We're going to look at water, and we're also going to review those organic compounds. Now, you saw water on the previous slide, and as we said, water is a molecule resulting from an oxygen atom sharing electrons with two hydrogen atoms. And water is a little bit unique because it is a polar compound. Oxygen is kind of like a big sumo wrestler, and the hydrogen ions would be like me and Mrs. Lambert, two little people. The oxygen atom pulls the electrons closer to itself. So if we were in a tug of war, the sumo wrestler would be pulling the electrons towards itself, and that gives the overall molecule partial charges. So water has partial negative charges on the oxygen and partial positive charges on the hydrogen. And that is what we call polar. There's charges on the molecule. And when we get more than one water molecule in an area, they tend to stick together. So water molecules are sticky because of hydrogen bonding. Because there's charges on these atoms, when we get two water molecules near each other, they're going to stick together. They're going to attract to each other. And this leads us to our properties of water. Water is sticky, and you should remember the words cohesion and adhesion. Water changes temperature slowly. So think about a pot boiling on the stove or your pool in the summer. It takes a really, really long time for that water to heat up. Ice floats, and if you remember my lovely picture of the ice and the fish underneath, remember if ice didn't float, we would have organisms kind of being pushed out of ponds and lakes in the winter. But instead, because ice floats, organisms can still survive underneath the ice when a pond or lake freezes. And lastly, water can mix with many substances. Now here's a quick, kind of easy way to remember the four. So if you're asked, what are the four properties of water? Think there are four S's. Sticky, slowly, this is kind of a stretch, but floats, and substances. So water sticks to itself and other things. Water changes temperature slowly. Ice floats. And water is really good at making solutions. Water is good with mixing with lots of different substances. And all of these properties help play a role in the human body and the environment. Now, the other reason that we talk about chemistry in biology is because chemistry makes up all of the molecules that make up living things. And you may remember the four: carbs, lipids, proteins, and nucleic acids. Again, kind of a silly way to help you remember this, but think CLAP. Carbs, lipids, nucleic acids, proteins. And if you make a big clap, think these molecules are big, like a big clap. Again, I know it's stupid and silly, but it may help you remember it. So macromolecules, macro means oxygen and two hydrogen. These molecules can be made up of hundreds or even thousands of atoms. And a couple things that you should be aware of when we're talking about these four different types is what is the main molecule, what are the building blocks, what do they do, and what do they look like. So for example, if you follow this chart across the row, carbs are made of monosaccharides. Remember, mono meaning one. When we put a bunch of monosaccharides together, we get a polysaccharide, meaning many, and a disaccharide, meaning two. The function of these, think about when you eat a carb, like bread or crackers or fruit or vegetables. Carbohydrates give your body energy. And to look at them, remember they kind of look like these little hexagon shapes or octagon shapes at times. And those are our carbohydrates. Lipids are typically made up of glycerols and fatty acids, so those would be the monomers. And the name of the polymer, the big molecule, could be either our triglycerides or triglycerol and phospholipids. Remember, lipids or fats are hydrophobic, so they don't mix very well with water, but they store lots and lots of energy. When we eat a fat, like the fatty part of bacon, that actually gives us our body energy. And any extra calories that we take in, our body will store that as lipids, as fat. And this is an example of one type of lipid. It's a phospholipid, and that makes up the cell membrane. Our third type, proteins, you guys should remember this from our genetics discussion. The monomer, or the building blocks of proteins, are amino acids, and the polymer, or the big molecule, is a polypeptide or a protein. The function of proteins, they do everything in the cell. Remember, this is how our genes are expressed. And to look at the molecular structure, this picture here is one amino acid. And the key thing about proteins, if you remember this, they have nitrogen in them. The rest of these don't have nitrogen. Lastly, nucleic acids, or DNA and RNA, you guys should also be pros with this, they are made up of those nucleotides, the phosphate, sugar, base molecule. We put a bunch of nucleotides together to get either DNA and RNA, and those two molecules are responsible for our genetic material. And from our genetics unit, you should remember that they both work together to help express genes, to help make proteins. And there's a picture of DNA and RNA. You guys should be pretty familiar with that. So when you're studying this slide, keep those things in mind. What's the name of the building block? What's the name of the big polymer? What do they do? And what do they look like? That'll help you study this slide. Our last thing that I'd like to talk about with organic molecules is how do we put these together and take them out? And if you guys remember, at the beginning of the year, we were building these different molecules. And every time you put two pieces together, you put two monomers together, you cut off a hydrogen and an OH group, and that pulled out a water. When we take two molecules and pull out the water, we call that dehydration synthesis. Here's a picture of that. On the right-hand side, we do the opposite to break down a molecule. We call hydrolysis. And if you look at this picture, we're taking something that's big, adding a water molecule back to it, and that's splitting it into two pieces. So dehydration synthesis, we're building. Hydrolysis, we're breaking down. Lastly, from our chemistry unit, we talked about enzymes. And enzymes are the little molecules that help to break stuff down or help with chemical reactions in a cell. And these enzymes help to get a reaction going and to help add the energy to it. Some specific terms. If you remember, the substrate is the thing that's being broken down, and it fits perfectly in the active site of the enzyme. It's like a puzzle. The substrate fits right in there, and the enzyme will help to break that down. So for example, in this picture, we have an enzyme. We have the substrate attaching to the enzyme, and the enzyme breaking down that into the product. So just review some of those terms for enzymes. As usual, if you have any questions or concerns, let me know, and we'll see you soon.